Okay, so in order to help segue, what I'm going to do is just give a couple impressions from what I've heard and also a couple of the details that I think haven't been mentioned, which can complexify life. So we started with the fact criteria, and that first makes things feel really controlled and really exciting, and we have a great definition. And then it took like less than 30 seconds to hear, well, you can't necessarily satisfy all factor criteria. And it's actually much more challenging to do this, which of course will inherently start a certain amount of disagreement as we then say, well, I satisfied 3, 2, so forth, depending on which region and so forth. But you can automatically see how some point of disagreement can start when you're trying to deal with a set of criteria that are foundational for the field, but at the same time, there's an ideal that we don't know how to fully get to. Then I think what we heard is a number of different perspectives of how to get there through different lenses, depending on one's expertise, the techniques being used, the perspectives of the brain, which makes sense because there are many perspectives of the brain, and each perspective, whether it be structural, functional, you go into looking at architectonics and so forth, every perspective has a broad range of methods that can be used. And then we all have to acknowledge each one of these methods is imperfect in some way or is sometimes poorly understood, hopefully less poorly over time. But then that brings us to fMRI, where when you look at fMRI, on the one hand, you can see some very ideal-looking things, like when Evan showed those maps, you get those beautiful transitions, which goes back to Alex Cohen's paper from 2008, which I thought was just a beautiful paper and always imprinted in my mind. On the other hand, think of what you've seen in the last couple of days when you start hearing things about propagation of dynamics across the brain, and the fact that even though on average you can get those perfect maps, if I actually were to selectively subsample particular states, suddenly I'm going to get very different looking maps. And it's not just awake versus asleep, it's actually while you're sitting there awake over time. So suddenly it starts becoming more complex, and then we start arguing, what do these variations mean? Are they about arousal? Are they about ongoing conscious thought? Are they about the task demands? And then it all becomes complex. And I just want to emphasize and close on that because I don't think that we necessarily have to get into this. Okay, it could be entertaining to watch the advertised boxing match. On the flip side, I think part of it is really thinking, how do you put the whole picture together? And I don't think anyone on the stage can actually do it yet. And I think that is the challenge for us all.
Yep. All right. Thanks. And so I guess we'll open up for a discussion. I think in terms of thinking about the discussion, I actually was not thinking of this as a boxing match. But I think I wrote consensus. I don't know if we'll reach consensus. I don't know if that's necessary. I think it might be also helpful to think about what are the differences. Are there differences between the viewpoints, and what are they exactly, so that we can maybe even do some hypothesis testing at some point. But anyway, with that, we'll open it up to the floor if anyone has any comments or questions.
Hi, I try to make it short as possible. It's rather than, it's more of a discussion rather than questions. So I'll point it out one by one. For example, like what is going on with one after the other, like methodology at this point. For example, while Harper oscillation was brutally targeted already, but in terms of the current resting state functional connectivity, defined the boundary, and we typically ignored it's a strong word, sorry about that. Ignored the fundamental characteristic of functional multiplicity while you generate an edge map, and when you consider about fundamental fact about different density of receptors or cell types or cyto architectures, they will generate different boundary. They should, but it is embedded in the one and only, for example, correlation matrix. And if you do not, for example, consider the high dimensional of the data, and that will give overlapping, consistent but questionable boundary location. And of course, the solution to that is so-called gradient manifold learning. But in terms of parcellation, the gradient was widely used. And in terms of the downstream analysis, especially in task or like case control FMI analysis, well, the Smith.
Why don't we let some of the people on the panel respond?
May I finish that? Well,
go ahead.
Okay, so well, these are the two spectrums, right? Like in terms of gradient, it's very important to define the precise boundary of a region of interest, which is proved already, like, well, in the simulated data by a paper 14 years ago by Stephen Smith. And in terms of global gradients, the method is necessarily smooth across the cortical area. But if the parcellation is based on that, the downstream analysis will be questionable.
It's just also to be fair to the other speakers in the room on the queue of questions
Yes,
behind
no
you.
problem.
We should keep our questions short.
It's not a question, it's a discussion.
Right, so,
Well-
yeah, so I mean, I'm kind of curious, Evan, I mean, what you think about that, like some of the stuff that Sarah was showing about, you know, the fact that the receptor distributions span, you know, these architectonic borders and partially, but not completely. Like, how do you think about that?
Yeah, I think this is the big question that so I don't think that any of our functional divisions have ever super nicely lined up with known architectonic borders, and we still don't understand why. I mean, we have these nice alignments in primary cortex, and then in association cortex, if you take brain networks, if you take parcels and you compare them against like the Ament's architectonic areas that she has so far, they don't line up super well, right? And I don't think it's clear why. I think we are missing something fundamental about the relationship between structure and function here. And I think that it's, my guess is that it's something confusing about the topographic organization, but that's only a guess, and that's sort of like a hypothesis that we're working with right now.
Yeah, I think I think it's going to come down to connectivity, but that's just my personal bias, right? That because connectivity also sometimes doesn't obey those clear cytoarchitectonic boundaries. In addition, the cytoarchitectonic boundaries themselves differ, right? Even in V1, you know, has the sharpest boundary of all, right? But if you, instead of considering layer 4, consider layers 5 and 6, like the boundary can be placed in a different location. And so I think that making headway on this problem, will require a deep dive into transition zones. And we should probably stop calling them transition zones because that makes them seem less important. I think that this will, like, my instinct is that the boundaries between the regions, because you can, look, I'm not gonna argue that there's not a difference between the middle of one area and the middle of another area in terms of how the cells are arranged, but there's like the transition zones and Nicola, it comes to that as well, to the receptor architecture as well. You said there are many receptors that then cut across areas and others will not. I think it's those, I do think if we could get a handle on what's going on in the transition zones, then we could get back to the functional, we could probably make the functional map. But I do want to press you on one thing, which is, you know, since you're here, you opened by saying, okay, an area is a sharp boundary in F, A, C, or T. But then over and over again, you fell back on, well, but this does not obey A. This does not obey A. This does not obey A. And so it seemed as though actually in your head, an area is defined by architecture. And that when function does not obey architecture, you think that's not an area. But why is that?
That's a great question. I've absolutely been wrestling with this for the last 10 years. You know, as I articulated, I do think that function has to arise from structure. And it could be the architecture, it could be the connectivity, right? But the reason that these things functionally differ is because there is something structurally different about them, is my perspective. But I also agree that this framework is not 100% consistent. I think that if we follow this framework, that like any difference in any one of the fact criteria, or even if we say like, oh, you know, function is special because it depends on the others, a difference in connectivity or architectonics, that counts as a different area, then we kind of have to think of M1 as being multiple different areas. I've been struggling with this a lot lately because M1 has different portions of M1 have differential connectivity to different portions of striatum, have differential connectivity. You know, the face area goes to cranial nerves, and the hand area is going all the way down the spinal cord. It's not completely internally consistent. Yeah.
The thing is, what's the definition? What is an area? And what you also said also was, you know, we know about the somatotopic organization in the motor and somatosensory cortex, retinotopic in the visual cortex. You're in V1 and everything's wonderful. You only have the blobs and interblobs. Not even V1 is homogeneous. You move into V2, you already start having problems, and also having problems between delineation correlation of cytoarchitectonically defined boundaries and retinotopically defined boundaries, and you're only in V2. And then you move up further into V3, V4, where you're starting to integrate more areas. So it's also, when you're looking at site architecture, the differences in site architecture between primary and secondary areas are so easy to see. But when you're moving into a higher association areas that have to communicate with many more other different types of areas across the brain, the differences in site architecture, the differences in receptor architecture get more subtle. And I think, for example, from the functional point of view, we need to design, it's very easy, well, not very easy, but it's very easy to design a task that just lights up V1, right? As opposed to everything else in the world, relatively. But try and identify or design a paradigm that will just light up, I don't know, I don't know.
14O.
Right.
14-0.
Well, we can do that for like the frontal eye fields, right? There are some of these areas that you can, pretty high up areas you can still distinctly activate.
Area 55B it's really like isolated from its neighbors, yeah.
Right.
But they are more like the exceptions that confirm the rule. So
right.
So, and I think that what we have to do is put all of these different maps all in a common space. And we've got to bridge the spatial scales of millimeters, micrometers, centimeters, and we've got to bridge the temporal scale that's provided by, my resting state post-mortem data, the eternal resting state, with up to the super spatial scales of EEG or whatever. And once we've managed to integrate all of those things, we'll understand it. But this is still the future.
I'll just add, I think that this has been particularly hard for the human brain especially, but even the macaque brain, compared to look at what I showed from the mouse, right? The ability to densely sample with methods that we feel really confident in, like track tracing, right? Which is more accurate than diffusion cryptography, like electrophysiology, right? How many intracranial, studies do we have? And they're all in diseased brains, and spatial transcriptomics. All of those have been really challenging, both in terms of ethics and in terms of the spatial scale. But we are tackling those, and I think that the next 2 decades will see those types of data become densely sampled across the human brain in ways that we haven't seen yet.
So moving to a question that was coming online, which was about, ties into what you're talking about. The question is, how do we decide if areas/gradients are true properties of the brain or statistical consequences of the methods we're using? Daniel, you perked up there.
No, I like seeing a punch thrown into the...
Well, let's be honest, when we just heard from Nicole, all that data, all the many levels, all the everything, that's like a massive statistical challenge and just fraught with potential for errors, let alone what we do at any one of those levels. So how do we move past that?
I don't know if I knew.
Someone have a statistician?
Yeah.
We need a set.
My hope is that, you know, convergence across different types of data, right? Like it's, hopefully if there's something screwy in the way we do stats with functional connectivity, the same screwy thing won't happen in the way you do you know, stats on track tracing, hopefully.
Okay.
So why don't we,
Yes, why don't we also move along the line just in order to get going?
Yes, thank you. I'm open for further discussion.
okay, thank you.
Hi guys, great discussion. It actually always, these discussions always harken back to when I was a young undergraduate student in the end of the 90s and everyone was talking about modularity. We wanted the brain to make these little modules that function. And
Steve Peterson hated that word.
yeah, listen, I don't disagree. And Brad Polster wrote a brilliant paper about working memory not being spatially located in the brain. And I thought it was a very fun foundational view when I was young. I'm going to shift a little bit and ask the question, okay, we're all trained in statistics, which means using group analysis, which means averaging things together. And this is near and dear to my heart, the idea of moving beyond the group averages. So I wonder how much of some of these struggles we have relates to our tendency to be trying to look at commonalities across people that blurs the functional topography, on top of which we're also trying to map on behavioral constructs that are based on our sort of psychological sense of what is a construct and not based on how the brain actually works. So kind of that idea of like how much is this driven by sort of a combination of group averaging versus not always well-defined behavioral construct using try to pull things apart in the brain, if that makes sense.
I think a lot of it. I'll say that from my own experience, when you know, 10-ish years ago, when we started moving from like looking at group average data to looking much more closely at really beautiful data in the individual, immediately the spatial scale for me of like the objects that I was looking at and thinking about shrank. Like everything became like, oh, there's this big blob, and that's the relevant you know, portion of the brain that's being activated. Now it's like small, punctate, different from its neighbor. It changed the way that I think about it, and I think that certainly it is a way that, because you know the real objects in the world are individual brains, and from my perspective, I think we have to be studying and looking at the individual brains to understand what the organization of the brain is.
I guess to go off that a little bit, also not just kind of spatial scales, but I think temporal scales, right? We often, you know, average over a bunch of trials from that one time point or however you do it across subjects. It might not just be the activity at that time point, but kind of as Mike said, you know, emergence of waves. So really it's the behaviors tied to the propagation of activity over a couple of seconds versus what it is over a second or whatever time scale you're on. So I think, you know, something not just spatial, but temporal, a similar idea too.
Cool, thanks.
Hi, I'm really enjoying this, but I'd like to kind of put a curmudgeon hat on and push back against it. So I think not represented in this room, I would say, is kind of like a hardcore primate electrophysiologist. And I think what they would maybe push back on this data is saying, you know, even if we had every map like we wanted, every scale of every possible amount of data, like the complete synaptome of the macaque brain, where would that really get us, right? I think some of these people would say, you know, a lot of the advances in circuit and systems neuroscience of the last few decades, like if you look at Krishna Shenoy and Mark Churchland's like M1 or premotor ideas, it wasn't from the fact that they better understood the connectivity even of individual neurons in M1, it was that they recorded a lot more of them and they analyzed the data in more complicated ways and found these like algorithms of how it was operating to serve a behavioral function. And so I'm curious both like what you think about that more functional idea, and also like even if you had the data at every level, the complete synaptic wiring and chemical diagram of your human brain, I mean, what would you do after that? I mean, what would that really tell you about brain function?
So I did do my PhD in primate electrophysiology, but I'm not sure that makes me hardcore. It's interesting you bring up, the BCI and Krishna's work because the M1 and the problems that they are tackling are very circumscribed compared to some of the other problems that we face, right? When we're thinking about emotion and higher order cognition and things like that. And so a big part of my research is focused on trying to identify neuromodulatory treatments for psychiatric disorders. And deep brain stimulation, for example, operates on white matter, and we don't know the organization of the white matter in the human brain. And even identifying that would be a major breakthrough in our ability to target it, along with the the technological breakthroughs that come with that. I wonder if underpinning your question is actually a deep one, because there's a lot that I would be able to do translationally, right, with the complete wiring diagram and receptor and cytoarchitecture maps of the brain. I think maybe underpinning your question is, who cares what the foundational organizational principle is? Who cares what the organizational principles are? And I think, again, that comes back to the type of problem that you're trying to ask. And the M1 BCI operates in a space where, the answer is maybe it doesn't really matter. But there are many, many other problems for which it certainly does.
Yeah, and I'm not saying certainly it doesn't. I'm just sort of, and an association cortex obviously is more complicated, but I think you could also argue that kind of at that level, perhaps that single neurons are so weird and their properties are so mixed in their selectivity that, there'll be one neuron out of 100 you could record from, and that does kind of the weird thing you were asking the animal to do, and not the others around it.
I think there's been a lot of progress on the mouse systems neuroscience side in what used to look like very, very heterogeneous structures that it turns out, when they're separated by their chemical and connectivity properties, start to look much more ordered, right? It's particularly in the amygdala, for example. I'm thinking of all of Kay Tai's work, a structure that seemed to just have neurons that did everything, right? Well, actually, that it's true, but those neurons are differentiable on the basis of their downstream targets. And so I think that some of the complexity that you're talking about would, we would be able to say more than, oh, it's just really complicated.
Moving to a question here that's on the app, says, we've heard a lot about areas and their boundaries, including sharp borders. At the same time, gradients are currently being discussed extensively. How do these two concepts fit together? Right, so this is the heart of one of these, some of the debates that the articles talked about is, are they compatible?
Yeah, I think they're compatible.
Okay, I guess we can move on.
Can you tell us how?
They're operating at a different spatial scale. Like, a gradient is a series of cortical areas sort of arranged in a line, and I don't think a gradient has to assume that there can be hard boundaried areas arranged in a sequential gradient.
I really think we agree on so much, Evan. I don't know how it ever took a weird turn, I would completely agree. I think the key question that this whole agenda has been shifting towards is not the question of sharpness of boundary, which is one that is rooted very much in the architectonic tradition and histology tradition, and is not necessarily optimal for addressing with MRI, I would say. I don't know if everyone, but yes, but the way in which we see these kind of macro scale patterns, and that can be captured by transitions between homogeneous. You okay, Tim? You upset? Oh, you got a line. Oh, okay.
I do think there's a testable hypothesis about every gradient or gradation. I think that Nicola is distinguishing between a gradation. There would be a macroscopic principle, but there could be sharp boundaries, right? So imagine a number line that goes 1, 3, 5, 7, 9 versus one that goes 1, 2, 3, 4, 5, 6, 7, 8, 9. So that's a gradation versus a gradient. And I think that for any given gradient or gradation any given macroscopic principle in the brain, that we should be able to figure that out. And that once again comes back to transition areas.
And if I may say one thing, if you're trying to do whole brain analysis, there's so much data there, right? That one of the first things that you have to do, as far as I know, is do like the dimension reduction, whatever, steps or, yeah? and if you're doing dimension reduction, what's it, the terminus technicus, well, whatever, then you're sort of like smoothing your data, or you're putting a filter on it.
Or you're losing information.
You're losing information, and that's why then we do have these 1, 5, 7, borders. We do have steps somewhere, but we're smoothing them out. That's why you're moving from gradations, which are also in the brain moving in different directions, and you're moving from, and then you get you're smoothing it out, and you're getting a gradient.
I think the metaphor that I've been using recently, and I'm still trying to develop it further, but is a rainbow, right? In which we have a long history of trying to figure out what are the colors of the rainbow, and where are the transitions, and how do we characterize something in which we could describe it as stepwise, but it also is continuous, and what are the tools that would allow us to differentiate that, and what is the most effective way to characterize it?
Sorry, when I've got my receptor autoradiographs, they're images, it's 8-bit images, so 256 gray values, a continuum, and the human eye just can't distinguish between these so well, so I divide them into 11 sectors, assign them colors, and suddenly, boom, the steps, pop out. But there are, as Sarah also said, when you're, the middle of one area, the middle of another area, there are gradual changes towards the border.
Now, I will say this. I think that as I've been looking more and more at really nice data in individuals, the more you see the gradient idea of running from primary to association cortex is broadly true, but there's many violations in its details, right? There's many sort of holes. Grattan's work calls it, variance, places where this relatively, stepped progression, there's a frank violation. There's a place in the middle of this progression which, looks completely different. And to me, I struggle a little bit to then translate that back to the original idea of these gradients resulting from the morphogenic gradients, right, in the protomap, because I'm not sure how that could produce these holes. Yeah.
Well, but so that sounds like what we're saying is that gradients are also imperfect, like what we said. The fact criteria also have their limitations, and that's an area where we could try to understand over time.
Let's move on here.
Okay, I'll try to keep it quick. First of all, also thanks for the refreshing session, which is why I'm allowing myself to ask a very naive question potentially. My background is engineering, so I'm more on the instrumentation and methods side in a sense, and in AI. And so using a very potentially simplifying example of artificial neural networks that everybody's working with right now, where we know the structure, I'm referring to what you said before. There might be something to solve between structure and function, and also subjects and group level similarities. So if you have a fixed architecture that you train with a random initialization in the deeper layers, feature representation can be very different, but the function can be very similar, right? The performance of the network could be very similar, although features are represented very differently. So I wonder if this can tell us something about, basically subject-dependent training of the brain. So if you have a structure that is globally comparable maybe, but the individual weights, the way that neurons have interconnected and are talking to each other are, highly individual. So maybe, I don't know if this question makes sense. I hope it does, and if it doesn't, then we skip to the next.
I don't know. I mean, it's a.
Should I rephrase it or should I skip? It's both possible.
Give us the simpler version of that
Okay,
question.
so
The non-engineering.
the simple version is artificial neural networks are not the brain. They're trying to copy some of its function. If you look at layers, they would basically represent the structure or try to approximate it. And in the deeper layers, which means not at the input, not at the output, you can have, let's say, your global optimization of getting, let's say, an image classified. Is this a dog or not, right? Might look very different in the inside. So the weights might be very different. But you get, have different networks. So the same architecture is trained multiple times, always tells you it's a dog. But in the middle layers, the weights, the synapses basically might look very differently.
So that's like the individual variability.
Right, and so what I'm wondering is if the structure is the same, but the function looks the same, but in between, this is where we would look at local connectivity maybe. It's different if this is telling us anything. But yeah. Okay.
It's of course it's a testable hypothesis. The observation that many people now have made is that the organization of the brain across individuals is broadly similar, but variable in its details across people. And I think it's unknown what consequences that have, right? It could be completely epiphenomenal. It could be that like these brains have exactly the same function that does not depend on the fact that this network is bigger or smaller, this network is shifted over a little bit in this person rather than that person, or it could be highly consequential, right? I think there, like the Lynch paper last year suggested that expansion of certain networks is related to vulnerability to depression. We don't know to what extent that's broadly true.
And I mean, maybe a little similar of a point, but if you were to just create a connectome from a random parcellation or a really fancy parcellation or something like that, you could probably still use that to train a pretty good prediction model of individual differences in behavior. So maybe the exact parcellation doesn't matter, at least when talking about various things, predictions across people. So kind of similar idea that, different ways of parcellating the brain or different ways of doing it, you might get the same predicted output.
So we're over, but maybe we'll do one more from Tim.
Well, I wanted to ask a concrete question that, I'm thinking a lot about individual variability, and that in the end we want to describe individuals. Do any of you have a particular view in the modality that you know the most about of an expectation that the number of objects that you're studying, by whatever means they would be the same in different individuals or not? Is there an expectation that the count, so to speak, of areas or whatever you're talking about would be the same from one person to the next? And if you don't think that's true, how would you then go about comparing across individuals?
So I think the number of areas is the same across individuals. It doesn't mean that their relative sizes are the same across individuals. So, we all have like a head and a face with two eyes and one nose and a mouth and everything, but the relative size of our nose or the position of our eyes relative to our nose does change between individuals. But we all have two eyes, one nose, one mouth.
And, I think if you're thinking about subaerial organization, there's plenty of examples where people differ, right? Like Glasser has Area 55B that in some people is just broken into two pieces. Doris Chow's work on face processing patches in the inferior temporal cortex. There's 6 of them, except that, some monkeys have 5 and some monkeys have 4, and one of them is like really big and maybe is 2 stuck together. They all seem to be able to recognize faces. I think there's something happening below the level of the aerial organization, I think, that is less pre-programmed and potentially more experience dependent and can vary a lot more across people.
This is where I would shift to thinking about it as a variation in a feature set. And so in a way what Nicola is expressing, that we each have certain features of a face, we can characterize the differences in those feature representations over space. And then get back to your question, how many dimensions are necessary to represent that? So I think that is precisely the open question.
Yeah, and one thing is, you think of cortical folding, you have variations in things like parasingulate sulcus, cross individuals, present asymmetries, and that all impact. So even if you had the same number of areas, as Nicole is saying, the relative representation is going to vary substantially in some portions of the brain.
And not even between individuals, left and right hemisphere in one individual.
Okay, I think we'll wrap it up there. Thank you, guys, for a wonderful discussion.
No blood.
And now we get to go discuss over beer.
Like the soft